Sometimes in science you feel like you're lost in a sea of acronyms, like NEB Q5 SDM Kit contains KLD mixture. Whoosh! And all of this is hiding some really cool science that allows you to make site-specific changes to DNA, to make specific changes to proteins that you want to study in the lab. And so this is a technique that we use for recombinant proteins. And so basically when we're making proteins in the lab, it's not something like CRISPR where we can actually use it to make changes inside of a living thing. We make changes in vitro and then we stick them in bacteria, and bacteria make the protein for us. And we can make changes to the DNA that has the instructions for making the protein, and then we can make changes to the protein that's made. And so we call this site-directed mutagenesis, or SDM, when we're making these specific changes to the DNA that has the instructions for making the protein, and in that way we're able to change the protein that gets made. So we can swap out a letter, we can take off a piece, all the various things that is then going to allow us to figure out how different parts of the protein help that protein work and what that protein does. So there are different ways that you can do site-directed mutagenesis, or SDM. So the technique that I use in my own lab is called SLICS, sequence and ligation independent cloning. There's quick change, and the one that I'm trying with my classes is this Q5 SDM kit from NEB. So let's go through all these acronyms. So NEB, that's just New England Biolabs. It's just the company that sells things. Q5, that's just the name of the proprietary polymerase, and so Q5 is apparently stands for quality, the 5 quality features that it says it has. And then the SDM, we already talked about that. That's the site-directed mutagenesis when we're making that specific change. And the KLD, that's part of the kit, and that contains the kinase, the ligase, and the Dpni. And so we'll talk about what each of these things do. And so first, let's talk about how this kit works overall. It is a PCR-based method in the beginning, and so PCR, polymerase chain reaction, basically make lots and lots of copies of a specific region of DNA, and you specify what region you want to make copies of using short pieces of DNA called primers. If you think about DNA, it's double-stranded, and PCR allows you to kind of heat the strands up to separate them, and then you can actually have the primers bind to those specific sites when the DNA is kind of separated. So we say the primer is like a needle, and then what happens is that the DNA polymerase that's in the PCR mix actually goes and extends off of the end of the primers. And so you have these short pieces of DNA that the DNA polymerase is going to start copying from. And so it's going to include that primer when it makes the copy. And if we have primers that have a mutation in them, so we make a change in the primer, well, now the piece of the DNA that's getting copied is going to have this kind of weird bit on the end, the mutated bit on the end. And so we make primers that have the mutation incorporated, and then we make copies of the rest of the template. And so the template is typically a plasmid, so a circular piece of DNA that contains the instructions for making a protein. And we're going to copy basically the whole plasmid, but we're going to do it in a way that the very small portion of the plasmid where we want to make the change, that's actually going to come from the primer and extending off of the primer and going all the way around. So I like to think of it a bit like you have a cookbook and it contains the secret recipe for these really, really great cookies. And your nemesis asks for the recipe for that cookie. And you're like, oh, sure, I'll give you the recipe. So what you're going to do is you're going to take the recipe from your cookbook and you're going to make copies of that recipe. But what you're going to do is you're actually going to change the word sugar for salt. And so now when your nemesis goes to make these cookies, they're going to be all salty instead of all sweet. And so you take kind of the first page and you make a mutant version of it. You change the wording in the version, and then you copy the rest of the pages, and they're all kind of like together. So you give this chapter, you make lots and lots of copies of this chapter, and you give it to your nemesis, and then they make lots and lots of copies of these really nasty cookies. And so in this case, we have these DNA primers that have the mutation incorporated, and then the DNA polymerase is going to copy the rest of the template that has the instructions for the protein. It's going to copy all the way around, and so all of this thing is going to get copied, but you're going to get this linear piece. And so you have one primer on the front and then one primer on the end, but the end is going to correspond directly to where that first primer starts. So these are blunt end primers. They basically, rather than overlapping one another, they have no complementarity to one another. And so instead, they're just kind of lining up back to back, and only one of them has the mutation. So this one has the mutation, say, and then this one is just kind of telling the polymerase, hey, stop here, because now we're going to incorporate this piece is the only piece that we're not making a copy of, and that's because that piece is coming from the primers. And so we have the primer that's going to then have the mutation. We build off that and we go all the way around. And we get this linear piece that we want to now join together. And there is an enzyme called DNA ligase that can actually stitch the ends together. But DNA ligase needs to have phosphorylated ends in order to do this. And so when we piece DNA letters together, nucleotides, so DNA polymerase pieces nucleotides together in the process of making long strands of DNA, and it does this using nucleotide triphosphates. So you have the nucleotide, so your DNA letters, with 3 phosphate groups, and the phosphate groups are actually providing the energy that the DNA polymerase needs in order to piece them together, because when you piece them together, you release phosphate groups, and then you get like energy that you can use to help power the process. DNA ligase needs phosphate groups too, but when you buy those primers, those short pieces of DNA, you typically don't buy them phosphorylated because they're going to charge you a bunch to phosphorylate them. And so you typically don't. And so the primers, though, are where you're going to have to join together, and they're not phosphorylated, and so we need to phosphorylate them. And the enzymes that phosphorylate things are called kinases. So the K in the KLT mixture is the kinase, and the L is the ligase. The kinase is going to phosphorylate the ends of the DNA so that the ligase can stitch them together. And so after you do your PCR and you make lots and lots of copies of the plasmid, but now containing the mutation, now you're going to need to stitch them together. You add this mixture, the KLD mixture, that contains the kinase, which phosphorylates the ends so that the ligase can stitch them together. Great, now we've got this circular piece of DNA. So why do we need that D for? Well, the D stands for DPN1. Now, DPN1 is a special type of restriction enzyme. And so restriction enzymes cut DNA based on specific features. And so often we think about restriction enzymes as like cutting specific sites on DNA based on their sequence. In the case of DPN1, it's a little different. And so DPN1 cuts up or like chews up methylated DNA. Now, when DNA is copied inside of bacteria, the bacteria methylate their DNA, or at least some strains of it. They methylate the DNA. And so they add methyl groups to their DNA. And that kind of has a few different functions, including allowing them to see which strand was old and which was new, and identifying it as their self and all this stuff. This enzyme, however, from this other bacteria says, okay, well, I see that you have methylated DNA. I know you're foreign. I chew you up. And so what we can do is we can add this DPN1, and it will chew up the parent plasmid, the template, because that template was made in bacteria. We typically use like a mini prep, so we grow up a lot of these copies of this plasmid in bacteria, and then we purify it out of the bacteria, and that's how we get our template. And so the template is going to be methylated because it was copied in the bacteria. But when we do the copying in a PCR machine, well, now we're not adding those methyl groups. And because we don't have those methyl groups, well, now it's going to be safe from chewing up from the DPN1. But the DPN1 is going to be able to chew up the parent plasmid. And so why do you care about this? Well, the parent plasmid, if you go now and you try to stick your mixture that has your mutated version and the small amount, but still some amount of your template, the original version, well, now the bacteria are going to take in both versions. And the version with the mutation, well, the version with the mutation might confer like a disadvantage to these bacteria. And so they're just going to, if you have like the normal, the wild type version, they might grow better. And so then you would actually end up with your colonies not even containing the plasmid that you want. So even though it might be a small amount compared to the amount of the mutated version that you have, if it has that growth advantage, you could end up with kind of having the bacteria transformed with, or basically the plasmid put in, that has the no mutation, which would be sad. And you wouldn't know until you sequence things, because what happens is that the way that we kind of tell whether the bacteria took in the plasmid is by using an antibiotic resistant selection method. And so on the plasmid that has the instructions for making the protein, you also have the instructions for making an antibiotic resistance gene. And so this might be a resistance to, say, ampicillin or kanamycin. Then what you do is, in the plates that have the food for the bacteria that you then basically, after you put the plasmid into the bacteria, so we call it transformation, you then put them onto plates. And when you put them onto plates, the plates contain LB, so bacteria like food, as well as the antibiotic that corresponds to the antibiotic resistance gene that's in the plasmid. The template has that antibiotic resistance gene. And so the template has it, the original has it, as well as the mutated version has it. So if the bacteria grow on the plate, it indicates that they took in a plasmid, but it doesn't tell us whether it was the mutated plasmid or the wild type plasmid, like the parent plasmid. But if we use DPN1, then we chew up the parent plasmid, and so then all of the colonies, so all of like the gross bacteria on our plate, should hopefully be our mutated version. However, we still want to go and send those for sequencing to make sure that it only has the mutation that we want and it does have the mutation that we want. And in order to do that, then we basically grow up a lot of these bacteria and we do one of those mini preps, like we did in the mini prep, to get the wild type template. And this, we send it off for sequencing, and hopefully we see that we have the mutation we want, we don't have any other mutations, and now we have this plasmid that is nice and pure, and we can stick it into cells that will actually make the protein for us, so expression cells, as opposed to the cells that we were using, the kind of cloning cells like DH5 alpha, which are really good at making lots of copies of the plasmid and making them like with high fidelity, so you make lots of copies of the exact same thing and you're not making extra mutations. But then when you want to go and make a protein, you want to stick them in bacterial cells that are really good for making protein, like BL21. And so, to review, Q5 is just the name of the polymerase, because you need the DNA polymerase in order to make the copies of the DNA. And when you make the copies of the DNA, you're going to incorporate a specific mutation that's incorporated into the primer to which the polymerase is going to extend from. The polymerase extends based on the template, so like the parent plasmid containing the instructions for the protein. It's going to make it all the way around, and meet up kind of where the original primer was. So one primer is going to kind of abut the other primer, but those primers aren't phosphorylated. So after you do all that copying with the PCR step, well, now what you need to do is you need to add a kinase. The kinase is going to phosphorylate the ends. Now the ends can be ligated or kind of stitched together by DNA ligase, and there's where you get your L in the KLD mixture. And the D, DPN1, is a restriction enzyme that cuts methylated DNA, such as the template DNA that you had to use as your, like the parent wild type non-mutated version that you were using in order to introduce the change. You want to chew up that template so that only the mutated version goes into the bacteria, and then the bacteria that grow will be able to grow because they have the antibiotic resistance gene that's also on the plasmid that has the instructions for making your protein, and the corresponding antibiotic is in the plate on which you put the bacteria with the plasmid that hopefully has your mutation, but you want to make sure by doing DNA sequencing.